Hi everybody, so this video we're looking at the t-test, which is a statistical test, and we're going to look at when we should use it, how to calculate it and what it tells us. So it's important that we know why we do a t-test. If you looked at the previous video about standard deviation, then you will have seen the example with our carrot farmer. So there's a farmer who wants to test two different fertilizers to see which one is going to give a better yield in terms of the length of her carrots. So she grows the carrots, she adds the two different fertilizers, one to each field, and then she takes a sample of the carrots and calculates the mean. And we do a t-test to see if there is a significant difference between the mean values, in this case, of the two fields. We can only do a t-test in certain situations. If you use a statistical test in the wrong situations, then the results will be meaningless. So in this situation, we've got two sets of data, Miracle-Gro and Growmore are the two fertilizers. And to use a t-test, you can only compare the means of two sets of data. We're also measuring the length of the carrots, and length is continuous, a continuous variable. So the data is continuous, it's a form of numbers. And that's the second thing which has to be true to be able to perform a t-test. Here is the data that was taken from the samples. If you plot that data, so if you take the height of, or the length, sorry, of the Miracle-Gro and you plot it against the frequency, then you should get a roughly normal distribution. And that's the third thing that you have to be able to do a t-test. Each data set has to show a roughly normal distribution. So we need continuous data, so numbers or percentages. We need normal distribution for both data sets, and we have to have less than 30 values for each of the two sets. If all of those are true, then we can do a t-test. So a bit like standard deviation, standard error, you don't have to remember the formula for calculating a t-test. This is what the formula is, you just have to remember how to use it. And what you can see is that we have our x bar, so this is our mean for data set 1, our mean for data set 2, and s is the standard deviation. So to do a t-test, you have to have calculated the standard deviation previously. And as I said, you can see how to do that in a different video. So we have calculated previously our standard deviations for both sets of data. So Miracle-Gro has a mean of 8 and a standard deviation of 2.20. Growmore has a mean of 6.5 centimeters, which would go up there, and then a standard deviation of 1.80. So you can see up here where we would plug those numbers in. So now we're just going to substitute them in for the equation. So we're just going to follow through, adding the numbers in, and then just doing each calculation bit by bit. So eventually you end up with a t-test value of 1.5. So what we have to do now is we have to take the t-value, we've just calculated it, and compare it to a value that you find in t-test tables, and we have to look at that comparison and we have to decide whether or not the difference between the means is significant or not. So when you look at a t-test table, so these are published tables which you can find in books on the internet, you'll see something like this. So we have something called the degrees of freedom, and then our critical value of t. So degrees of freedom, this is how you calculate it. It's just a way of including the sample size into your statistics. You don't have to understand why we do that, but generally bigger samples tend to give more reliable data than smaller samples. But the calculation of the t-test needs to know what the sample size is. So to calculate degrees of freedom, you just take the number of samples in the first data set, add to the number in the second data set, and minus 2. So we have 8 samples, 8 carrots in each data set, so our degrees of freedom is 14. So if you look at the table, you can see here that there are different numbers of degrees of freedom, and a real table would go all the way from 1 all the way down to about 30. You have to know where in that table to look. And then in the critical value of t, there would be also 3 different columns. And we have to look at this one here where it says p equals 0.05. In biology, we always look at that column. In physics, you might use this column here, but in biology, this is the only one that we care about. So when you look at the table, you'll see values here. These are the critical values of t. And again, you have to know where to look in that table so you can compare the t value you have calculated against one of these. So because we've worked out that we've got 14 degrees of freedom, and we know that we need to look at the p equals 0.05 value, that means that this is the critical value of t, which is important for us, 2.15. Okay, so if our critical value of t is 2.15, what does that tell us? If our t value is less than the critical value, and our critical value is 2.15, at p equals 0.5, so at this probability level, another way of saying that is if t is less than the critical value at a 5% confidence level, in that case, there is no significant difference between the mean values. Now, this is important. This word here, significant, is really important. It's highly likely that there is a difference in the means because we're looking at, in this case, living organisms. We have variation between individuals. It's highly likely that the mean values for our 2 data sets are going to be different. But whether or not it's significant is the important thing. So do we think that the difference is being caused by something biological? In this case, if t is less than the critical value, then no, we don't think there's any significant difference. There's no reason for the difference, and so any difference between the means is just due to chance, just due to the random variation between individuals. Now, our p-value is just a way of saying how confident we are in that result. So we use a p as 0.05 or a 5% confidence level. If you were to use p equals 0.01 or a 1% confidence level, that's what that means, then what you're saying is we are even more confident that there's no significant difference. So as you go this way down the table, you're basically more confident in your results. So we just have to quote this number here. Okay, what about if we have a different critical value? So if we did our t-test and we found that actually our t-value that we calculated is greater than the critical value, which is 2.15, at p equals 0.05, then in that case, we would say that there is a significant difference between the mean values. So there's a difference and it's significant, so we think that there is a reason for that difference. There's something causing it. It's not just due to chance. In this case, we could also say that we reject the null hypothesis. The null hypothesis is what you write at the beginning of an experiment, and the null hypothesis is basically saying that we don't think there is anything going on here. We don't think that there will be a significant difference. So the null hypothesis says there's no significant difference, so we have just found there is a significant difference, so we can reject the null hypothesis. So let's just put that in the context of our carrot farmers. So here's what we did. We wanted to find out if there's a significant difference between the means for our 2 different fertilizers. We looked at our t-value against our critical value, and we found, if you remember, a t-test value of 1.5. That's what we calculated a little while ago. So we can say that the t-test value of 1.5 is lower than the critical value of 2.15 at a probability level of p equals 0.05. Therefore, we can conclude that there is no significant difference between the mean values. And in this case, we mean the mean length of carrots. What's important here is that we actually specify what we mean. So there's no significant difference between the mean length of carrots, and then really importantly, any difference is due to chance. So the mean value of Miracle-Gro and Growmore was not the same, but that difference is not significant. The difference in the means is just due to chance. If we had done it and we got a different result, so just let's suppose that we got a result when we did our t-test of 3.6, then our conclusion would say this: it's higher than the critical value, there is a significant difference, and we would then get rid of that last section. So this, what you can see here now, this would be our conclusion if we had calculated a t-value which was higher than the critical value. Okay, you should have everything you need there to be able to use t-test effectively. Thank you very much. That's all.